In today's video, we're going to cover part 1 of chapter 2. So this is all about elicited behavior, habituation, and sensitization, or at least how much of it we can cover in this part. Now we're finally going to get into some of the real learning part of this course. First chapter I find is a lot of background and terminology and stuff, and while we're still going to have tons and tons of terminology, we actually get into some of the really cool stuff that makes this course really fun. So today's lecture, I've also posted a bunch of YouTube videos on eClass, so I won't be showing them in the lecture because those belong to somebody else, so I can't post slides or post videos with other people's videos in them, but I've included links in a couple of places, including in the slides, and then direct links on our eClass site, so you can follow along. So pause this and go watch those or whatever, but they're just really neat, really quick videos that help illustrate what I'm talking about, and sometimes that helps flesh out the concepts we're discussing. So lots of videos, lots of cool ones. A couple with warnings, just in case you don't like watching things like rats in operant boxes reacting to loud noises. If there's ever anything that people might be uncomfortable with, I try and put a flag just to let you know what the video is about, so you can know if you want to watch it or not. And if you don't want to watch it, I have a description there, so you kind of get the basic idea. But that was a very, very long introduction for this video. So our basic outline, this is what's going to be covered all throughout chapter 2. This video is only really going to get up to about here, but this is the whole setup. So this chapter is focused on how elicited behavior can be modified by experience through the processes of habituation and sensitization. So elicited behaviors are really any behavior that is made to happen by some kind of discrete stimulus. And this sort of simple setup and our simple learning that we're going to see with both sensitization and habituation end up being the most basic unit of learning, and we're going to kind of build from there. So we're starting with the simplest components, and then we're going to work up and build up with more complex interactions as time goes on in this course. But it's kind of where we're starting out. So we're going to look at what elicited behavior is, what effects of repeated stimulation will have on that elicited behavior. We're going to look at some of the theories behind habituation and sensitization, and then how that can kind of connect into more complex things like emotions and motivated behavior. So that's where we're working towards. But just to get started, I kind of want to do a reminder back to Psych 104, where you would have been taught in the learning chapter about the big three of learning. We have habituation, classical conditioning, and instrumental or operant conditioning. So we have event-alone learning, event-event learning, and behavior-event learning. So we're going to be basically covering the number 1, habituation, and some of the other stuff that involves habituation or is related to habituation, because, of course, the story you were told in 104 wasn't the complete picture. So we're going to build on this as time goes on in this class, but just a little bit of a roadmap so you remember where we're coming from. And as a quick reminder of that, for habituation is animals cease to respond to some kind of repeated event. So this is that single event learning. You learn about one particular stimulus, and there isn't necessarily any generalization to other stimuli. So you could habituate to a loud sound, but that doesn't really mean you've habituated to a bright light or even to different sounds. And so there's a link here that shows the habituation of a startle response in a rat, where basically the rat's initial response to a very loud sound is to jump or start. And as that sound gets played over and over and over again, the response becomes less and less and less. So we can say that the rat has habituated to that sound because they have learned not to respond to that sound. For classical, or sometimes called Pavlovian conditioning, that's when we're looking at relations between events that help us learn something about those events. So event-event learning, where the presentation of a previously neutral stimulus, something like a bell, becomes associated with an unconditioned stimulus, something that you have an automatic response to, something like food, and that previously neutral stimulus becomes a conditioned stimulus that you've learned to associate with that unconditioned stimulus, leading that conditioned stimulus to produce the same kind of response as your unconditioned stimulus would. So the idea of a bell making you salivate because you've learned to associate the bell with food, and food makes you salivate. So Pavlov's standard conditioning and learning there. And so there's a cute little video of this idea of conditioning your roommate. So basically just a guy with one of those staples that was easy buttons, and he hits the button, shoots his roommate with a Nerf gun, and repeats until his roommate learns to associate that sound with getting shot with a Nerf gun. And then oh no, I have forgotten to change the title here. There we go. Fixed with the magic of editing. So for operant, or instrumental conditioning, because all of these have two names, because why not? This is where you have new behaviors are formed, specifically with goal-directed behavior. So this is behavior event learning. So you learn that by performing a particular behavior, you can cause a certain consequence to happen. And when that consequence happens, if it's a good thing, it'll make you do the behavior more. If it's a bad thing, you will do the behavior less. So you've learned something about doing a particular behavior to achieve some kind of outcome. And so another video here, this is the idea of shaping a lever press response. To learn to press a lever, and pressing a lever will eventually get, I think it's rats, so it's probably pressing a lever to get food, and that would be an operant conditioning setup. So you can check out that one. And so that refresher and some fun little videos leads us to the actual topic for today, which, to start off, is elicited behavior. And as I've kind of hinted at already, elicited behavior is behavior that occurs in response to some environmental stimuli, some kind of event that's happening in our environment. And when we're talking about behaviors, environmental stimuli just means like whatever's around us or whatever's even in us that's causing us to do something, or that's sending a signal that leads to us doing something. So eventually we'll talk about sort of a mental environment, so things that we're thinking about. For elicited behaviors, we can think of a more behaviorist approach, where an environmental stimuli is going to be something like a smell in our environment, a sound in our environment, something that's immediately surrounding us. But this idea of elicited behavior is actually a little bit simpler than what we usually think of when we think of learning. So usually when you start thinking learning, you think of maybe deliberate instruction, like sitting in a class, having someone lecture to you, going to lessons where someone shows you how to do something, places where you would practice, like learning an instrument or something like that. We think about how that learning is going to be taking place and probably being shaped by things that we've had experience with in the past, and what we're going to be trying to do with it in the future, and, you know, that more complicated view of learning. But as I said in our introduction here, we're starting with the simplest of concepts, where we have to acknowledge that behavior isn't infinitely flexible. Organisms are born with pre-existing behavior systems and tendencies that are going to constrain how our learning occurs and what changes we can expect from that training. No matter how hard you try, you can't teach a fish how to fly. the textbook uses kind of an interesting comparison that I hadn't really thought of, and this is the idea of sculpting wood, where as you're learning something, you kind of have to work within the constraints of the material that you're given. So if you're a wood carver, maybe your sculpture has to be shaped based on the size of the piece that you're working with, any flaws or knots within that piece, the direction and the density of the wood grain. So you have to work within that pre-existing structure, and you're going to see the most successful outcomes when you take into account that sort of pre-existing situation. So if we then apply that to learning, learning is going to be most successful when it works within sort of those biological constraints of an organism. So if you want to work on birds, because that's my go-to example always, you can focus on behaviors that come naturally to birds. And so we see lots of stuff looking at how pigeons peck things, especially if they're being rewarded with food, which we'll talk about at length later on, because for pigeons, pecking is a thing they do when they want food or when they're getting food, so they work together naturally. So it goes much smoother when you take into account these constrictions, these biological limitations, and work within them. And so I guess the most basic of restrictions that we have are sort of innate reactions that happen in response to our environment. So all animals, even single-celled organisms, all the way up to humans and other complex animals, we're going to react to events in our environment. And we have some reactions that occur almost innately without having to be learned. So some examples of things that happen without our necessarily learning to do that, or things like that. Let's not get caught up on saying the same phrases over and over again. So an example would be our pupils automatically constrict when we're exposed to bright light. We have an automatic withdrawal reflex if we touch something that causes us pain, like something that's very, very hot. We'll sneeze if a particle of dust enters our nose, or we'll sort of scrunch up our eyes if something touches our eyelashes. I've skipped over the food in your mouth elicits salivation because off the top of my head, I'm not sure if that's an innate thing or if there is learning involved in that. But that might be something to consider. But the simplest form, because I said we're starting simple and working our way up, the simplest form even of elicited behavior is something like a reflexive behavior, where we have an eliciting stimulus that causes an innate linked response. So in this example here, we have a puff of eye causes an eye blink response. So if somebody blows air in your eyes, you're going to close your eyes. If you've ever gone and had your eyes checked, if they've done that glaucoma test where they blow a puff of air into your eyes to see how your eye reacts, same exact situation. And you don't have to learn to do that. That is something that is going to happen reflexively or on its own. So you don't have to learn a reflex. It's going to happen naturally. These can be modified by learning, but we will get to that. Now, this is a diagram that you're probably familiar with. This is the most basic diagram of a reflex arc. So if we have something that is reflexive, it's a behavior that's going to happen without brain involvement or without direct brain involvement. So you might have seen this as part of a diagram showing somebody touching something hot and withdrawing their hand from it. We'll talk in just a minute about your knee-jerk response. They're all kind of the basic same setup. It is good to note that this is an example shown in humans, and it tends to apply to most vertebrates, but this is specifically a diagram from humans, so just keep that in mind, that some other groups will have a different setup. But in humans, a simple reflex arc will involve as few as 3 neurons. So we have our sensory neuron that's coming in from some kind of sensory receptor, whether that be temperature sensor on our hand, or a pressure sensor in our skin somewhere, or whatever kind of sensor that's bringing in information that's going to travel into our central nervous system. So this is our spinal cord. And then there's an interneuron, where it's going to be making the connection between that incoming information and then the outgoing command, basically. So the interneuron takes that information from our input with the sensory neuron, going to be making a sort of shortcut decision here, and then there's an outgoing sort of command along the motor neuron, and that motor neuron connects to the muscles that are going to cause whatever the reaction is going to be. Now, you might remember that in this setup here, there might be a signal that transmits up the spinal cord to the brain to let the brain know what we were sensing and what we did in response, but the brain isn't involved in making an actual decision on how to respond because it's a reflex. It's something that's going to be happening very quickly and pretty much automatically. Now, doing it quickly is really useful because if it's something, say, that's causing you pain, immediately pulling your hand back from something that hurts you is biologically beneficial because it means you're not being hurt for as long. A fast response is helpful. And so a lot of the things that we look at for reflexes, it is biologically beneficial for that response to happen quickly. The trade-off for that very quick response is that we're very set or restricted in how this response is carried out because we only have 3 neurons involved and they only connect in a very specific way that is hardwired and results in very stereotyped behavior. So you're going to pull your hand back in the same manner, whether you're touching a very hot surface or maybe something that hurts you. It's all going to kind of work the same way. You're going to pull back, and you don't really get any say in how that behavior happens. Now, for a specific example, because we've talked about you touch something that's too hot or you touch something that hurts you, we can also look at the knee-jerk reflex. So this is when I've never actually seen this done in person. I've only seen it done in movies and TV shows, but you go to a doctor's office, you sit on the exam table, they take out their little rubber mallet, and they tap the tendon just below your kneecap, the patellar tendon right here. And the correct response to that is for your muscles, specifically your quadriceps, to contract, and it causes your leg to kick upward or outward. And so we can see the same basic diagram here, but set up to work with this reflex arc where we have a sensory nerve coming up from the tendon. In the spinal cord, we have a connection here. So there would be an interneuron mediating that communication between our sensory nerve's input and then the motor nerve with that output. And the motor nerve connects to the muscle in our quadriceps and causes that reaction. So a nice, simple reflex. And no matter how many times they're going to hit that part on your knee, that knee-jerk reaction is going to look fairly similar. Like it's following a very set path, it's a rigid response. And different people will all have the same kind of reaction because it's a biological reflex. So if these elicited behaviors are so simple, so basic, why are they, what are they useful for? Why do we use them for? Why do we have them? And I've kind of hinted at this already, and it's the idea that these reflexes are adaptive. There's some kind of biological benefit here. It helps with fitness or survival or whatever. It's something that is innately useful or beneficial, and so it could have evolved and adapted within that species. So it's contributing to an animal's well-being or survival, it's increasing our fitness, and therefore it's allowed to evolve in the population. So a lot of our reflexes can also be considered adaptations because they've occurred and adapted within our environment. So some of the examples that we have here, an eye blink reflex, which is kind of like a puff of air in your eye. So there's a video of a dog where basically tapping close to a dog's eye, not necessarily on it or like touching any part of it specifically, tapping near the eye causes the dog to blink. We also have a withdrawal reflex, a gag reflex. We see head turning in infants and respiratory occlusion reflex in infants. So there's a lot of these are, again, biologically useful. So for the gag reflex, it helps prevent choking. So things that cause us to gag are things that our bodies might choke on. So it is helpful to try and stop that from happening, hence the reflex. Head turning in infants actually helps them feed because they're going to turn its head towards what might be a way of receiving milk. This respiratory occlusion reflex in infants is actually a really neat one. And for whatever reason, I didn't know about this one until fairly recently. I guess once I started teaching about learning. But if an infant ever has restricted airflow, so if they have, say, a blanket over their face, or if they have trouble breathing, they have this reflex where they pull their head back, they move their hands, and they'll try and cry. So there's sort of a set of events that all happen together. And so the idea here is by moving your head, you might move away from something that's covering where you're breathing from. Moving your hands can help pull things away from your face, and crying could expel something that's blocking your airway. So all of those work together and could help almost like troubleshooting problems with breathing. So that's a really neat one. And like I said, all of these are reflexes. They're all hardwired. They happen innately. So that's really, really neat. So with some of those examples of simple reflexes, some of them you can see across multiple groups of animals, things like a startle response or our pupils reacting to bright light. Those tend to be consistent across multiple groups. Other elicited behaviors might only be really prominent in one species or a small related group. So example, you'll see suckling in mammals because that's the only group that really suckles. But there's some variation there. Our next sort of variant upon reflexes is basically like a little bit more complex than just a simple reflex. And these are called modal action patterns, or MAPs. These tend to fall into that second category where they're species typical or small group typical. And they're not just a single reflex, but they tend to be sort of a group of associated patterns or instincts. So they're still genetically programmed. They're still going to be happening innately. They're something that happens without training. But there may be a couple more steps than just a reflex. And so these MAPs, these combined patterns of behaviors, we can see them in a bunch of different species for things like feeding behavior, sexual behavior, territory defense, and even catching prey. One example is that in male ringdoves, they're going to choose a nest site and build it cooperatively with a female, so they won't start building a nest until they have a female, whereas male sticklebacks will build a nest and then court females. And they can't skip that nest building stage, so they can't start courting until they've accomplished that first stage. So there's a very fixed pattern of how that works. And with all of these different kinds of MAPs, there's a threshold for the eliciting stimulus, whatever stimulus it is that will cause that behavior to happen. The threshold can vary. So in that nest building idea, if it's the right time of year and the male is in a particular sexual state, then it would be easy for them to start that behavior to build the nest and start courting a female. But if it's the wrong time of year, or if they're not healthy enough, or if they're not sexually mature, then that threshold might be harder to hit because they haven't sort of gotten to the right place for that to exist. So these thresholds aren't constant and aren't sort of held at the same level all the time. There's other stuff that's going on there. So again, our modal action patterns, though they're related to our reflexes, they are already showing us that they're just a little bit more complex. And we're going to talk about these for a little while. So the first of the very famous modal action patterns is the egg rolling response that you tend to see in geese. So this was a study that was done a very, very long time ago, back in the 30s. So this was Lorenz and Tinbergen. And there's a video of this, so you can click on that link, watch it on the e-class or whatever. But the basic premise is that in geese, they have this modal action pattern where if they see an egg outside of the nest, they will follow a very strict set of actions to bring that egg back into the nest. So if we look at the steps on the diagram here, the first step is to extend their neck, to then extend to the point where they're touching the egg. They scoop their beak under the egg, and what they do is they kind of nudge it back towards them into the nest. And so they will scoot that egg underneath their bodies back into the nest. And this series of behaviors, the extension, the wrapping around it, and the scooting it back under their bodies, is all very rigid and structured. So in this example, our eliciting stimulus, the thing that triggers this modal action pattern, is the egg outside of the nest area. And there's lots of stories about discovering this and sort of testing the extent to which this behavior will happen, where if the goose sees that egg outside of the nest and starts the first step of this modal action pattern, as soon as they start extending their neck, they have committed to completing that whole series of events. So even if you reached in after this stage and took the egg away, that goose is still going to reach to the spot where the egg was, scoop their beak underneath where it had been, and roll, now nothing, back towards the nest. They're going to complete that entire set of events no matter what, now that they've committed to. So it's really interesting that once these modal action patterns have started, they're going to keep happening to completion. Now our next example here looks at another map, this one being sort of a feeding response in gulls. So here we have an adult gull, and at the bottom of their beak they have this red spot. And the idea is that when their chick is young and still in the nest and needs to be fed, the chick will peck at that red spot, and that triggers the parent to regurgitate food and feed the chick. Now, for this situation, the first question that researchers would ask would be, which part of this situation is our sign stimulus? Sometimes this is called a releasing stimulus, but it's asking which feature is essential to this map happening. And in this situation here in the wild, we're not quite sure what it is, right? Because it could be the red spot. It could be the beak itself. It could be the shape of the whole head. So we have to pick it apart and see which aspect of this system is our sign stimulus, which is essential for this particular response. And so for the basics of this experiment, we're really just going to look at this diagram over here, but this would be, do chicks peck at a fake head? So if they have a head of a bird, plus the beak, plus the red dot, so they've got basically everything they'd have in nature, as expected, they have a certain amount of responding. So I think they've scaled this to 100% responding to the most natural of their stimuli. So that's our baseline because that's what they do in the wild. The next thing they tried was, okay, let's remove the rest of the head. We're going to effectively just have a beak with a little bit of white, but the beak still has a red spot on it. How much responding or how much pecking do we see from the chicks? And the answer is almost the same amount as the original stimulus. So this is still eliciting quite a bit of responding, lots of beak pecking. So a beak with a dot is sufficient. The next attempt, or the next variant they did, is they removed that red spot under the beak. They kept the rest of the head, but removed the red spot, and we see this dramatic drop-off in responding. So by removing that red spot, they can see that reaction, that response, doesn't happen. So that gives us some indication that the red portion is essential to this whole modal action pattern. So that's telling us that maybe the red is involved in our sign stimulus, and maybe it itself is the sign stimulus. So the next thing that they checked was basically just a red stick. So here we have a red stick. It's pointed at one end. They had it oriented in the same sort of direction as the beak, so it's pointing downward. And you can see that there are some bands of different colors here. So there's a contrasting red thing towards the tip. And we see not only similar responding to the original natural stimulus, but even more responding than that. So in this case, this stimulus, this red stick, is sufficient to trigger that modal action pattern. So this tells us that the yellow coloration on the natural beak, the shape and color of the head, and they also even looked at noises that the gull parents produce. None of those are required to elicit this pecking behavior. All that you need is a long, thin object that moves, that has a contrasting red patch near the tip. That is sufficient to release this modal action pattern. So that's our sign stimulus here. And because we're actually seeing more responding to this stimulus than even to the natural stimulus, we might call this a supernormal stimulus, an exaggerated sign stimulus, because it's producing more responding, and it's an artificial stimulus. So that kind of fits the bill here. So if we want to look at supernormal stimuli, we can look at other experiments where they manipulate the natural stimuli, and if we exaggerate a certain aspect of that stimulus, does it produce increased responding? So if we have eggs, so these eggs were used to test that egg retrieval behavior, this time in gulls, not in geese, but same basic idea. And the way we can read this diagram here is that, so A, B, and so on, these are all different types of modifications that they would have done. And then they have at the bottom modal value. So the idea is that the ones on the left-hand side produce less responding than the ones on the right-hand side. So increasing effectiveness as you move to the right side of the screen. And so we can look at, they can be either brown or green. So these ones at the bottom are green, everything else is brown, and they're giving us an idea of size and shape, and if they're speckled or not. And so we can look at this here, and we can see that if we look, let's say along here, so we're saying that all of these have about the same effectiveness. So it doesn't matter if it's green, or if it's brown, or if it has spots, a little bit if it's green without brown without spots, because that seems to be a little bit less effective. But up here, they have something that isn't even the right shape. They have sort of a roundish square that's the right color and has spots and is kind of the right size, so it's effective enough. So it tells us that the shape and the color and even the spottedness isn't necessarily affecting the responding as much as we might expect. And the factor that they end up finding that is sort of most responsible for the driving this behavior, deriving this map, is actually the size. So if it's the right color and it's the right shape and it has the speckles, so it's kind of ticked all the boxes where it fits all of that, the larger that stimulus is, the more responding they get. So simply by making that egg bigger, we can call it a supernormal stimulus, and it's causing an exaggerated or more intense response. I find it just interesting to look at all of the different things that they tried, because they would have done multiple trials with each of these different models that they designed to check a different thing, because they wanted to look at colors and spottiness and shape and size and all of these different effects. So this was a very thorough experiment. But even within the different types, so even if we only look at the green eggs, larger is still better. Even if we look at our rectangles, larger is still better. So that size seems to be driving this relationship in a really cool way. As I guess, a different example of supernormal stimuli, we can look at sticklebacks. And these guys are probably something you've heard of before, because they are the example for supernormal stimuli and modal action patterns, at least when you don't want to look only at eggs and birds. And these guys are a great way to show us, again, that the most realistic model is not always the one that's going to elicit the largest response. So to get started, we need a little bit of background on sticklebacks. Sticklebacks are fish, and male sticklebacks tend to guard their nest and territory in a very aggressive way. And their acts of aggression, sort of the stances that they take and their aggressive behavior, follows a modal action pattern. So they have a very predictable pattern of responding to things that they view as a threat. And in this experiment, they have another set of models that they put together. And so they have a fish-looking one, and then they have all of these strange shapes where some of them kind of look like a stickleback, but without the fins. And some of them are just like blocks of wood that are painted red on the bottom. So kind of squarish, too fat, too thin. And all of these end up eliciting that aggressive response from male sticklebacks. So the shape itself doesn't really matter. So if we want to try and understand why this might happen in terms of that adaptive significance and how this might have evolved to be, it might just be that it's better to attack anything red. So the fish would be better off wasting the energy to attack something that's red on the off chance that it's a competing male versus the penalty that they would suffer from not attacking. Because if a male comes into your territory and you don't attack and chase him off, you can lose fitness to that male. So if you're protecting a territory in a nest, maybe another male comes in and mates with females in your area, in your territory. So there's a pretty strong penalty to not chasing off a potential competing male. So wasting the energy on attacking anything that's red on the bottom might have been evolutionarily worthwhile, just in terms of trade-offs. So that's another interesting example, and there's a video that goes with this as well. And in the natural world, because these modal action patterns exist, we actually see naturally occurring exploitation of these maps. So the example here is that you have cuckoos, this big guy on the right, who are, cuckoos are basically brood parasites or nest parasites. So the parents would come in and lay eggs in a nest of another bird. And they really don't care what bird it is, because they'll lay the eggs in that pre-existing nest and they'll leave. And they leave whatever bird's nest that is to care for their young. And so this baby cuckoo will beg for and end up receiving food from this foster bird, which is clearly not even the same species. So you can have much smaller birds who work themselves into the ground trying to take care of this larger bird that isn't even their offspring. And the reason why this might be working is because it's exploiting that pre-existing map. So like we said, there's lots of feeding behaviors that are tied into sort of naturally occurring patterns. And in this case, the sign stimulus would be a red gaping mouth and the begging calls that go with it. So we might even look at this as a supernormal stimulus because their beak is larger. So it's a bigger stimulus, it's more red, and maybe because they're bigger, they're even louder. So this would even lead these parents to feed the cuckoo more than their own offspring just because that stimulus is more intense and would trigger that modal action pattern more often. So a really interesting way of making use of or exploiting that existing behavior. And we already kind of talked about the threshold for the stimuli to elicit these behaviors, and the fact that that threshold can change depending on the state of the animal. So I'll just do this really, really quickly. But within a daily cycle, whether you're hungry or full will affect whether you engage in that behavior or not. So an example is a full baby gull won't peck the red spot on its parent's beak because it doesn't need the food. So the threshold to initiate that MAP isn't there or is much higher, so they don't do it as often because their state at that time means they don't need it. We can have seasonal cycles where it's breeding season. So maybe fighting off an invader in your territory only happens when it's near breeding season, but if it's not breeding season, you won't expend that extra effort. We also have things like called a lunar cycle here, but more like a monthly cycle. And that would be like a reproductive cycle in humans where their current state, especially in human females, can increase preference for different things, especially in how they view males and their attractiveness. So there's lots of really cool research into stuff like this where we have these nice repeating cycles. So those are a bunch of like very specific MAP stimuli, but what about other eliciting stimuli? We can talk about lots and lots of studies that have shown predisposition to identify different kinds of stimuli. So humans are really good at detecting snakes or things that are shaped like snakes, and we'll identify them and react to them much faster than we'd react to something like flowers or frogs, something that's non-threatening. And we even see this in some of our closest ancestors. So other apes will have that same faster response to something like a snake versus something neutral like a flower. So it tells us that that's something that's probably passed down in our genes. It gives us this biological preparedness, which is really, really cool. But how do we get from those very simple, very basic, innate collections of actions? How do we go from just a reflex or just a modal action pattern? How does that work into our actual behavior in the real world? How do we organize our behaviors? How do we put it all together? So in nature, our behaviors aren't isolated. Our individual actions tend to be organized into sort of functionally effective behavior sequences. So this is what we're going to be looking at here, are these behavior sequences. And again, these tend to be species typical or groups of species typical. But these behavior sequences tend to kind of cluster together or consider together different behaviors that all work towards the same common goal. So the example we're going to use here is a squirrel trying to find food. So they're going to look around for food sources. They're going to find a tree that tends to have nuts on it. They could climb that tree, start reaching for nuts. Then they have to crack the shell, get the meat out, chew the nut, swallow it, and so on. So all of those behaviors, while being sort of their own behaviors, their own distinct units of behavior, work together into that umbrella of finding and getting food. And so with that example, we can look at appetitive behaviors and then the following consumatory behaviors. So if we're looking for food, the early on behaviors are appetitive behaviors. They're behaviors that brings an animal into contact with some kind of stimulus that will release the later consumatory behaviors. So in our example of finding food, the early behaviors are going to get them to the point where they have food in their hands or paws, as the case may be. So these appetitive behaviors tend to be early on in the sequence. The stuff like looking around, finding the right tree, and getting into that tree. And because this is happening early on, there tends to be a little bit more wiggle room here. There's a lot less stereotypy here. So searching for the right tree or how to get up that tree or something like that, it tends to be less rigid and controlled by just your genetics and just your innate behaviors. So maybe we see some shaping with learning. Maybe through experience, you've learned which kinds of trees tend to have nuts. So that falls under this appetitive behavior. And once you get to that point where you have come into contact with a stimulus that triggers the next step, that triggers our consumatory behaviors, we move on to those consumatory behaviors. These are the later part of the sequence. This is, you have a nut. You're going to crack the shell. You're going to pull out the meat. You're going to chew it and swallow it, and so on. And so these end behaviors occur late in the sequence after you've encountered that stimulus that triggers them, and they tend to be highly stereotyped. So they tend to have a very specific eliciting stimulus. So maybe a squirrel has a particular response to acorns versus walnuts versus hazelnuts versus whatever. And so they're going to have a different set of behaviors that happen pretty much automatically, because they're a lot more stereotyped at this stage, that allows them to effectively get into that specific kind of nut. Now that I've gone through the example, this is probably a good point to tell you that this idea of appetitive and consumatory doesn't mean that this only applies to food, searching, and eating. These terms are actually adapted or borrowed from ethology, where they had been looking at sort of foraging behaviors, but we're using these terms not just for food, but just to mean early and late behavior. So it doesn't have to just be foraging. Foraging just happens to be one of the easiest ways to visualize this. And so to go over that example again, we have our early appetitive behaviors, would fall under this like general search mode where it's non-directed, it's a lot less stereotyped, the behaviors are a little bit looser. So things like general search or even a focal search of once you're in the tree looking for those nuts. Our focal search is sort of ending our appetitive stage and then transitioning us over to that consumatory phase where you're looking for one specific thing in your localized environment, and once you find it, you launch into handling and ingestion, our consumatory phases. So that's the foraging side of things here. A non-foraging example would be getting into a fight. So if you're, I don't know, in high school, people would get into fights all the time, depending on what school you were from, I guess. But the initial phase of the fight where people are sort of threatening each other, the early stages, what we could call our appetitive behaviors at the very beginning, trying to egg somebody on, insulting them, that sort of thing, that's going to be very different depending on who it is that's fighting. And they're going to be a little bit more freeform and able to say whatever works for them. But once people actually start fighting, once you get into the hitting someone, that part tends to be a little bit more stereotyped, where you react to avoid getting hit and you lash out in a pretty stereotyped way. So we could call that a consumatory behavior because it's a lot more stereotyped and it's kind of towards the end of that involvement. And that doesn't have to just be humans fighting, that could be fish defending their territory or whatever. We can look at appetitive and consumatory just as replacements for early or late behaviors based on how they're being framed. All right, so that was the elicited stimuli in a nutshell, going off of squirrels, I guess. Now we can actually get into how do we change or adjust some of those elicited stimuli? How do we get into things like habituation and sensitization? So here we're looking at how our elicited stimuli, or our elicited responses, can change with repeated stimulation. And you might remember that Descartes had this idea that reflex responses are very rigid and structured and unchanging. So he thought that all reflexes are going to occur the same way every single time. This is not true. Though reflexes are fairly stereotyped and they are fairly consistent, repeated stimulation can alter that behavioral response. So through the idea of either habituation or sensitization, our reflexive responses can change. So even though we've looked at it as very stable, elicited behavior can be subject to modification through experience. And so we can look at either increasing with sensitization or decreasing with habituation our response to different stimuli. So habituation, as I just said, is when you decrease that response intensity with repeated exposure to a stimulus. So this is just a still frame, but there's a video up on eClass of a rat habituating to a very loud sound. And it's a very simple video. The idea is there's a very loud sound, and anything that's startling in your environment will cause a startle response. So the first time the sound happens, the rat jumps. The second time the sound happens, they jump. The 50th time the sound happens, they don't jump as much. The 100th time the sound happens, the rat barely reacts at all. That reduction in responding, that reduced startle response, is the process of habituation. So once they reduce their responding, we can say that they're habituating to that particular situation. So let's look at a couple of experimental examples of elicited behavior and this repeated stimulation. So we're going to do 3 different examples. We're going to look at salivation and taste ratings in humans, visual attention in human infants, and then startle response in rats. So the first one here is salivation and taste rating in people. In this experiment here by Epstein and colleagues, they gave subjects either lemon or lime juice. And our black lines here are looking at their measured saliva output over trials. So how much did their mouth water when they were given a taste of either lemon or lime juice? And then on this side, we have their ratings of how pleasant the taste is. So if we look at trial number 1, this point here and this point here are both trial number 1. So the first time they were given a taste of, let's use lemon juice for our example. So first time they're given lemon juice, they salivate quite a bit. They rate it as sort of unpleasant. That's what high on this scale means. And that's, lemon juice can be jarring, so it kind of makes sense. If we jump ahead and look at the 5th trial, so this is the 5th point here, we've noticed that our amount of saliva produced has dramatically dropped off, and our rating of how unpleasant it is has also dropped off. So here we're seeing a reduced response and a reduced perception of it over time. And so after 10 trials, the amount of salivating they're doing is much, much lower. And after 10 trials, their rating of how gross it is has gotten much lower as well. So they don't mind it as much. So here we can say that the individuals have habituated to that particular taste. Now the cool part here is that we see these dots standing off on their own. This is for an 11th trial, and this is where they did something slightly different. So if flavor A was, as I said in my example, a lemon juice, then for this 11th trial, they would use flavor B, the lime juice. And this shows us that even though they had just habituated to lemon juice, presentation of the exact same amount of lime juice showed almost the same kind of saliva response and almost the same rating of unpleasantness as initially experienced with the lemon juice. So even though they're similar, we can see that they habituated specifically to lemon juice, and lime juice wasn't habituated to at all because it's new and different. And so they would have done this counterbalanced. So half the participants would have lemon juice as flavor A and lime as flavor B, and the other half would have lime as flavor A and lemon as flavor B, but everybody showed the same basic pattern. And so, like I said, this is giving us two things. The one that I focused on already, that response decrease is specific to the stimulus that they were habituated to. So it was specific to lime juice and not lemon juice. But also it shows us something even simpler than that our elicited behavior, that salivary response, isn't fixed. Even though it does happen naturally, it's elicited by our environment and we don't necessarily control it, it can be changed with experience, which is really, really cool. Oh, and apparently I had included an extra graphic showing that these were a different flavor, but we already talked about that without the extra lines, so we can skip that slide. Our next example, as promised, is visual attention in infants, in this case, human infants. So they used a task of human-infant visual fixation, which is what we see over here, where you have a baby and you show them a particular stimulus. And what we're measuring is how long they would pay attention to that particular stimulus. So we would call that fixation time, which is a fancy way of saying how long will they stare at that stimulus before they get bored. Now, this is a great place to comment on the fact that this method here of showing them a stimulus and measuring how long they will pay attention to it is a really great way to test subjects that can't talk. So we're using it for human infants, but this works for any other animal that can't tell us what they're seeing. So we're measuring their ability to pay attention or how long this holds their attention, and we can then compare two different stimuli and see how their sort of holding of attention varies between the two. So in this experiment, they have a 12 by 12 checkerboard stimulus and a 4 by 4 stimulus. So one is more visually complex than the other. And then we can use that idea of stimulus specificity that we just talked about with our lemon and lime juice example, and we can see how long they stare at one versus the other. So if I hop back here, we can look at this graph showing how long they stared at either of these stimuli over number of trials. So let's start with our 4 by 4 stimulus. So this one here, the large checkerboard, and we start on trial 1. They stare at it for about 5 seconds, and then they look away. The second trial, they stare for less, third less, and over time less and less until eventually they're effectively bored of that stimulus. They look away very quickly. So this gradual decline in that looking behavior or that fixation tells us that they have habituated to this stimulus. So kind of in line with what we would expect. With our 12 by 12 stimulus, this more complex stimulus here with our open circle line, we actually see something different. Where on the first trial, they stare about the same amount that they did with the 4 by 4 stimulus. They're pretty close together there. But on our second trial, we actually see more staring. So in the second exposure to this busy checkerboard pattern, they stared at it for even longer. And in fact, it took, I guess, let's say the 6th trial before they were back to looking at it for just as long as the first time around. So in this case, they looked a lot more to this stimulus. So that isn't habituation. That's actually our second idea we're going to talk about in just a minute, which is the idea of sensitization. They're paying more attention to it. So instead of having a reduced response, like we saw with our 4 by 4 stimulus, we now have an increased response, which we're going to call sensitization because they're responding more. All right. Now we can use a very similar setup, that idea of looking time, to pick apart different stimuli. So here we can have human faces that we can show to infants, and based on how much we know about faces, if we give them time to habituate to a face, maybe over time they will become habituated, because that's what we're aiming for in this experiment. They'll become habituated to that particular face. And so what we're testing here is if we habituate them to a particular face and then show them either a new face or a familiar face, can they tell the difference? So we're using that habituation and that reduced looking time as a measure of, is this a stimulus they're familiar with, or is it something new that they're going to look at for longer? So it's a kind of way around the fact that we can't actually ask infants if this is familiar or not, so we can use habituation to see if they recognize it as something they've seen before and have habituated to, or if it's something new and they're going to respond more to. And in this really cool study that I kind of wish we had more time to talk about, they found that not only were babies able to habituate to this face and then maintain that habituation, but only with the familiar face. They responded differently to novel faces. They were able to do this even when they looked at the faces from a different angle. So if they were trained on faces that were looking straight on and then were tested with faces from the side, they were still able to recognize which ones they had seen before and which ones were new, which is super, super cool. But yes, I love some of these experiments. They're really neat. And then for our third and final example here, we're going to talk about the startle response. So we've already kind of introduced this idea of rats habituating to a very loud sound. So now we're going to look at the data that goes along with that video. And again, there's links on here to see that video. And there's a couple that go together with this experiment. So they're all labeled the same way, but we can talk through what the data shows us. The first two graphs here are actually showing us something very similar. The first is long-term habituation, and the second is short-term habituation, which is basically how often are they exposed to that really loud sound. And so in this one here, they're only getting exposure to that really loud sound once a day. So at the beginning, on day 1, they hear it once. On day 2, they hear it once, and so on and so forth. So we still see that downward trend that we now know to associate with habituation, where the magnitude of their startle response is decreasing with time, so they are slowly habituating to that particular stimulus. However, if we look at this short-term habituation, our stimulus presentation is down to every 3 seconds. And so instead of this being a measure of sort of days, with once a day being presented, these are showing us blocks of 30 tones at a time. So if we're doing a stimulus every 3 seconds and we're doing 30 tones, this is over the course of 90 seconds. And so at the beginning, we do see sort of that more responding, but these are like the scale here is the same for startle magnitude across all 3 of these graphs. They're just separated out. So even in our first block of tones, even in the first 90 seconds, our responding is much lower than what we saw in the long-term habituation, because even in that first block, they were exposed to the tone every 3 seconds. So there's tons of exposure there already. But we still see that downward trend where over time, they're showing less and less of a response. But because they're getting so much more exposure in such a tight time frame, our overall rate of responding gets down to almost no response, almost no startle response at all. The cool part that we're going to learn new here with this example is that after a period of rest, we see something called spontaneous recovery, which is even though we've gone through this habituation and they have this learned lack of response to the sound, if we give them a break and give them that new stimulus again, we see an increase, a spontaneous recovery of responding, where this responding returns even after habituation. So because of this break period, they kind of go back up to higher startle magnitude, a more intense response than what we had seen previously because of that break. And so this is really important to know because while we can modify that startle response, while we can modify those reflexes, they don't completely go away. So it's not like you're unlearning them, it's just that you're learning something else. And so we'll talk more about that later. But I think that's enough background for part 1 of this lecture. We'll do part 2 on Thursday.